Next up, I'd like to share with you our speaker, Sarah Rijkenberg, who is a doctoral student in the Applied Bioscience program at Ontario Tech University. Sarah's presentation today is titled Go Fish, Using Plankton Characteristics to Monitor Ontario's Fish Populations.
Here in Ontario, we have more than 250,000 lakes, which provide us with numerous ecosystem services. One of the industries that profits the most from these lakes is recreational fishing, which brings in over $2 billion annually. The Ontario government does monitoring work on these lakes to ensure there's no environmental degradation, but also to make sure that the fish populations are maintaining healthy numbers. It's crucial that we do this sampling work to ensure there's enough fish present to actually keep that industry going. While doing this work is important, actually getting samples from hundreds of lakes is extremely expensive and time-consuming. Anyone who's ever gone fishing for a day can tell you how long it takes to catch just one fish. Now imagine trying to catch hundreds. It takes a huge amount of equipment and teams of people to operate it. As well, some methods use mesh netting, which can take multiple days to complete and can harm the animals that they catch. As a result of these factors, only a small fraction of lakes are actually getting sampled each year. And the reality is, most lakes only get sampled a few times a decade. And this leads to a massive lack in management information. But what if I told you there was a better way? Something that was cheaper, faster, and that requires less manpower. I want to talk to you today about plankton. Plankton are microscopic organisms that float in the water and make up the bottom of the food web. In essence, plankton is fish food. We can look at the amount of plankton present for fish to eat, but we can also look at the type, because plankton comes in a really wide range of shapes and sizes, and this affects how nutritious it is for the fish to eat. The idea is that we can look at a sample of plankton and it will tell us what the fish population looks like as a result. All it takes to do this is a jar of water and one person on a microscope. Now, you still need someone who's pretty knowledgeable about plankton to do this kind of work, and these experts can be somewhat difficult to come by. But that's where I come in. I want to create high-throughput plankton monitoring programs. I'm working with a machine called the FlowCam. You can pour a sample of water directly into the FlowCam, where it takes thousands of pictures of every particle that it sees, and simply gives the user that data. However, we don't have an algorithm that's good enough yet to fully remove a human from this process. But I think I can teach the FlowCam how. Imagine just one jar of water being able to tell us what an entire population of fish looks like. This would revolutionize the way that we study food webs today. I hope to make that a reality. Thank you.